Hello everyone. Today's concept is about the mutation and how mutations takes place. What is mutation exactly, and how it would be applicable to know the different kinds of phenomena in case of cancers, or how a disease get expressed due to these mutations. So first of all, we should know what is mutation. Uh, Any sudden change on the base pair of DNA we can refer to as mutation. Obviously, mutation is due to so many external agents or the factors, and basically the term which we use to express the causes of mutation is called the mutagen. So the chemical which are responsible to cause the concept of mutation on DNA, especially that is mutagen. And there are so many kinds of mutations. So depending upon whether it occurs in the chromosome, chromosomal mutation, might be the structure of chromosome, might be uh, the uh, the number of chromosomes. So mutations may be expressed either on the change on the base pair of DNA or change on the single base on the DNA, the so-called point mutations. So other than that, mutations may be the frame shift mutations, and how that frame shift mutations can occur on an living cell within the DNA. We are able to express it by taking an example. It is very easily expressed in NCRT book by taking an example. Ram has a red cap, so taking this RAM has red a cap. So we are knowing the genetic codes are made up of three bases, and that three bases here can be designated as RAM has a red cap. And eventually these mutations, it may be deletions or insertions. So sometimes certain single base come on the DNA region, DNA base pair, and when that DNA get inserted, I mean the bases get inserted on the DNA, then that is called insertional mutation. Sometimes it is called deletion, sometimes it is referred to as transversion, translocations, as in the case of chromosomal mutation. So, if we are going to that sentence, just counting Ram has Z cap, taking this C alphabet as the genetic code, and eventually what happens if Ram has Z cap and Ram has big red cap. So here big word, this is again another new word which get inserted one by one. At first B comes, then I comes, and then G comes. So what would be the final sentence now when the big is added on that very genetic code sequence, taking as Ram has a big red cap. So obviously, taking the three alphabets of a genetic code, we are able to determine the final genetic code, and obviously the frame of the reading on that very sentence Ram has a big red cap. It will be altered. So obviously, when we count the genetic code from forward to the backward directions or reversely, the pattern of the genetic code so form after insertion of the word big one by one as B, I, G, the total reading frame will be shift, and the conclusion will be whatever the previous reading frame before addition of the big word, the reading frame was different. But when the insertion of this big word takes place, then the reading frame fully change, and that's how the genetic code would be changed, and that's how the sequence of the amino acid which would be determined, that would be also changed. So taking this example of uh, Ram has big red cap, we are able to get changes on the gene sequence or genetic code, and thereby the change in the sequence of the protein. Similarly, suppose now we are going to delete the word B, one by one, B, I, G, and there may be red also. So when we are going to delete each alphabet from that sentence, uh, Ram has big red cap. we are going to delete the word big and red, obviously the sequence of the genetic frame will shift, and that's how the genetic code and their sequence would be different, the amino acid synthesis or their formation would be different, and that's how the total reading frame would be altered, and we are able to know how the mutations takes place due to either single base change or the double base or maybe a total genetic code. Obviously, mutation and this mutation very familiarly we are able to use certain mutants of Drosophila during the study of linkage and crossing over, and we are very finely can express how these mutants are expressed in their different generation like F1 and F2, and how we are able to find out the recombinant types and parental types due to these mutations. Mutations also determine certain genetic disorders also. Sometimes some mutations are very fatal. In the case of human being, there are some disease like sickle cell anemia, thalassemia. All these are due to the gene mutations which are actually determining the phenomena of this disease. One more metabolic disorders in human, that is called phenylketonuria. It is actually the disease where the phenylalanine is actually not able to convert into tyrosine due to the absence of the enzyme phenylalanine oxidase. And if this phenylalanine oxidase is the enzyme is not functional, the phenylalanine pyruvate actually is accumulated in the brain regions, and thereby the mental retardations and the loss of hair. All these are the symptoms of the phenylketonuria. So the thing is that mutation actually takes place not only during the developmental stage, certain genetic switches become on and off. So uh, the normal one, the switch which are maintained, the mutation processes going on during developmental stage, and that is very systematic and in a pattern way. But suppose these mutations are not actually helpful for cell metabolism or cell disorders would takes place, then it may be fatal as we are knowing. Certain mutants are there which are due to, mutagens are there which are due to certain rays, and rays have become again ionic radiations and non-ionic radiations. So certain chemicals are they are just like a picric acid, which is actually used in the laboratory to conclude certain lab experiments. So we must take care of certain chemicals when we are using in our naked hand. We must cover our hand with the gloves, because certain chemicals have the tendency when it comes directly in contact with our skin, the cell of our skin get mutated and thereby may cause a very severe conditions later on. As we are knowing, the UV rays which are coming to the planet Earth, and it is falling on our eyes. So the cells of our retina get mutated, DNA mutations will take place, and it may or it might have a cause of xeroderma pigmentosum. So these are little bit idea about mutations. So in next topic we'll discuss the different kinds of mutations, and this is the basic idea on this. So thank you. Next time.